So we're going to talk a little bit about the structure of the cell now, and also how this relates to metabolism. So we're going to be talking about human cells, largely in our metabolism lectures, and these are what we call eukaryotic cells. So this means that the cells we'll be looking at have multiple compartments, and this is because we have these organelles. So we have a membrane enclosed nucleus and also other cellular organelles that are enclosed by a membrane. So things like the mitochondria. They're also a lot larger than our prokaryotic cells, which are our bacterial cells. So other hallmarks of the prokaryotic cell is that there's actually no nucleus. The nuclear material is actually within the cell itself. So we have a single DNA molecule in the center of the cell, but it's not enclosed in a nucleus. And the other important thing to remember about our prokaryotic cell is that there's no membrane bound organelles. So key things for us to remember about our eukaryotic cells is that we do have our DNA present in the nucleus, and also we do have the other cellular organelles which are membrane bound. So here we have a picture of our eukaryotic cell. So around the outside of the cell we've got our plasma membrane, which is another word for cell membrane. So remember we saw in our lipid lecture that the cell membrane is actually made up of what we call a lipid bilayer. We have the polar heads facing the inside and the outside of the cell, and then the non-polar fatty acid tails actually extend inwards and create a non-polar environment in the center. So this is actually useful for not allowing polar components through the membrane. So if we want to get a polar component through the membrane, we need a specialized protein called a channel. So you can think about the main function of the plasma membrane as basically just a barrier between the inside and the outside of the cell. So you can imagine on this side of the cell we have the inside, and on this side is the outside of the cell. So it's just this barrier that is preventing everything that's outside coming in and everything that's inside going out. So this is basically what holds the whole cell together, and it allows it to exist as the cellular entity. So we have the inside of the cell being represented here as the cytosol. So the cytosol is the liquid component of the inner part of the cell, and within the inner part of the cell we also have our membrane bound organelles, so things like our mitochondria. We've also got our endoplasmic reticulum where the ribosomes are attached here. Remember, the ribosomes are the site of protein synthesis. We've also got lysosomes up here. So we deliver specific components of the cell to the lysosome when we want to break them down. So for example, if a protein has passed its use-by date and we want to destroy it, we can actually take it to the lysosome to break it down. And of course, in the center of the cell here, we've got the nucleus. So what is the main function of the nucleus? What does it contain? So remember, we have our genetic material within the nucleus. This is our DNA, which contains all the instructions for how the cell needs to function to keep the organism alive, essentially. So the mitochondria are really important for metabolism. We can think of these as the power stations of our cells, and these are the part of the cells that are responsible for generating our cellular energy in the form of ATP. So the mitochondria themselves have this double membrane where they have both an inner and an outer membrane. And between this inner and outer membrane, we have what we call an intermembrane space. So we've got our mitochondria. This looks something like this, where we've got our outer membrane, and then on the inside of that, you've got this ruffled membrane in the center, and this membrane is actually called the inner membrane. So the space between these two membranes, that's what we're calling the intermembrane space. So we'll talk about the mitochondria more in later lectures because it's extremely important for ATP generation, and the locations within the mitochondria actually have different roles in the ATP generation. For example, the proteins that are part of the electron transport chain are actually embedded into this inner membrane. So we've got the enzymes, ATP synthase complex, and also these cristae as part of the inner mitochondrial membrane. And these are all important for, so these enzymes and the ATP synthase complex are important for generating ATP. So within this ruffled membrane that I've drawn, we call these the cristae, and we actually have proteins embedded in these that perform particular functions. For example, ATP synthase is used to generate ATP. So we actually have the ATP synthase complex coupled to the electron transport chain, and through a process we call oxidative phosphorylation, we're able to generate ATP. Alright, so here we can see a bit of a nicer picture of our mitochondria. So we can see the spaces that are created by the ruffling of this membrane. We call these the cristae. And the space between these two membranes is what we call the intermembrane space. And the membrane around the outside of the mitochondria is just what we call the outer membrane. So we can see the representation of some of the proteins that are actually embedded within the inner membrane. So one of these is actually the ATP synthase complex that we talked about earlier. So we also have the electron transport chain complexes, which are also proteins, and these will be embedded in this inner membrane as well. So you can see a high resolution picture of our mitochondria on the right hand here, stained in green. So we can see the lines here, which actually represent the folding of that inner membrane and the formation of these cristae. Alright, so we're now going to have a look at ATP in a bit more detail, and also some of the other very important components of our metabolic pathways. So you can see here the coenzymes that I was referring to earlier. So we're going to have a look at each of these structures in detail. So all of these ones that I've circled in purple here are what we call the coenzymes. So what is a coenzyme required for, remembering back to the previous session? So we require a coenzyme to associate with an apoenzyme to be functional. So this is what we call when an enzyme is a hollow enzyme, it's actually got two parts. So another name for a hollow enzyme is a conjugate enzyme. Alright, so we've got our hollow enzyme, which is a combination of the apo enzyme. So remember, this is the protein part. All enzymes are proteins, at least the majority of them are. And then we've got also the coenzyme that is required. So depending what the hollow enzyme is, it will have a particular apoenzyme and coenzyme that associate together to form that active enzyme. So remember, it's only when both of these components are together that we are functional. So on the left-hand side here, this represents the functional enzyme. So each of the components on the right-hand side here, the apoenzyme and the coenzyme, are not functional alone. They need to combine together to create the hollow enzyme to be functional. So the coenzymes that we see here, FAD, NAD, and CoA, are used in a variety of reactions where we combine those with an apoenzyme to facilitate the chemical reaction. So we'll see that these are extensively used in our metabolic processes. For example, in glycolysis, we actually require NAD at one of the glycolysis steps to bind to its apoenzyme to facilitate that reaction. So we'll see here that each of these coenzymes and also ATP are what we call nucleotide containing compounds. So remembering back to our nucleotide lecture, the nucleotide contains three components. It contains a sugar, which is either going to be a ribose or a deoxyribose, and that sugar is actually linked to two other components. We have the phosphate on one side, and on the other side we have our nitrogen containing base. So down here, the nitrogen containing base for all these compounds is actually adenine. So when we have our nitrogen containing base being adenine, and then we connect that to a ribose sugar, and then we've got a single phosphate connected, then that's what we call adenosine monophosphate. So we can actually turn this into a diphosphate by attaching another phosphate to the phosphate end here. So because we have two phosphates, we call this a diphosphate. And then by fusing another phosphate onto the end here, we've now created a triphosphate or three-phosphate compound. And this is actually what we have when we form ATP. We have the adenine nitrogen containing base, the ribose sugar, and then we've got three phosphates attached at the end here. So the phosphates at the end here is actually the part that is the most energy rich. So we can actually release energy by breaking the phosphate bonds. So the bonds that are holding the phosphates together. So that's how we derive energy from our ATP. It's how we release energy. So we can see we've also got our cyclic adenosine monophosphate. So that's just a variant of our monophosphate up the top here, but instead of being a straight chain, we actually form a cyclic structure, so a closed circle. Whereas all of these other ones are just straight chains, so adenosine monophosphate, straight chain, diphosphate, straight chain, and triphosphate is also a straight chain, but we can actually have the monophosphate adopting this cyclic-like structure. So we can see how we actually need to build up to ATP. So we can actually attach a phosphate to the AMP, or adenosine monophosphate, to create ADP. So this is our variant that has our two phosphates. And then if we want to get up to the triphosphate, we need to add an extra phosphate to get to ATP. So this is actually what we do in the oxidative phosphorylation step. We actually have ADP and we're adding a phosphate onto that, and as well we have to provide energy, and by providing the phosphate and the energy in combination with ADP, we're actually able to form our ATP, or adenosine triphosphate. So this is the process that we do right at the end of our metabolic pathway, where we use the electron and hydrogen rich NADH and FADH2 to feed into the electron transport chain, which links in with oxidative phosphorylation to create our ATP. So we'll see a lot more detail about that later, but just to be aware that this is sort of the ultimate goal of our metabolic processes, to make ATP in this way. So we talked a little bit about our B vitamins in the previous lecture. So the B vitamins are actually vital to use our carbohydrates, our lipids, and also our proteins as an energy source. So we do need to keep the levels of these B vitamins up through our intake. Although we don't need a huge amount of them, if we starve our body of these vitamins over a long period of time, it's going to be very detrimental to our metabolism and the ability to derive nutrition from our proteins, carbohydrates, and also the lipids. So we can see that each of our different B vitamins actually is involved with forming a different coenzyme. So the ones that we'll be talking about most are our niacin, which is used in making the NAD, and also the riboflavin, which is used to make FAD. We'll also be talking a fair bit about CoA, which uses pantothenic acid. So you can see that there are a few other ones here also, but the three that I've circled are the ones that are the most important, and we're going to be referring to these throughout the lectures on metabolism.